Welcome. I'm very excited to start this little series of new webinars concentrating on our Biodiversity Beyond National Jurisdiction Treaty. So today we're actually going to have an opportunity to discuss the section of this treaty which deals with marine genetic resources. And we have a fantastic lineup of speakers who will give us a bit of an introduction to this whole theme. After that, we will also have 45 minutes of opportunities for our attendees to give a presentation on a series of questions that you will pose. So please take this opportunity to actually go straight into asking questions that you might have in your mind, and we will select those. We are looking at quite a few people. We had over 250 sign up for this event, and this is the first time we're trying out this GoToWebinar software. So we might have some glitches, and so unfortunately that might cause a little bit of delay or confusion here and there. So bear with us. You are guinea pigs on this one, but we're optimistic that we will be able to do it. So why are we organizing this set of meetings? Well, we wanted to have an opportunity for the negotiators who are working on the treaty and who will be having further discussions soon on marine genetic resources to really discuss these type of topics. We also wanted to get an opportunity to a little bit of a broader community to ask questions and get a bit of information of where we are. Now, if this turns out to be useful, if there's a demand for it, we're also happy to try to organize other types of meetings of this nature, either in this type of format or slightly different formats depending on what the needs might be. So what is it in the Marine Genetic Resource Treaty that we need to address? I think there's a number of questions that are on many delegates' minds, and those are the types of questions that we're trying to understand. So the first one is, of course, how are we doing with science and actually helping science through this new treaty? How can we get and foster a better collaboration between countries, between scientists, and between the different groups that are really trying to learn and deepen our understanding of the open ocean, but perhaps also find new opportunities for us to actually benefit the life of mankind. And here, I'm particularly happy to say we have a fantastic set of speakers who have a lot of knowledge and experience on this topic. And fortunately, we've been able to assemble people from pretty much all the major oceans around the world. So we're optimistic that we will cover quite a few different aspects of what needs to be included in this marine genetic resource part of this treaty. Now, in order to get this to work, we thought we would start with our first speaker, and this is Harriet Harden Davis. She is a Nippon Foundation Ocean Nexus Fellow at the University of Wollongong in Australia. She studies high seas governance and is a guest investigator at Woods Hole's Oceanographic Institute at this morning, leading their work on Deep Ocean Stewardship Initiative. So without further ado, Harriet, you have the floor. Oh, you're still muted, Harriet. We need to get you unmuted. You need to unmute yourself, okay? Does it work? Doesn't work, okay. Let's try one more time. All right.
I'm now unmuted, thank you. I believe the slides are coming online as well from the head office over there. Let me use this moment to say thank you very much, Carl Gustav, and thank you. Good morning, good afternoon, good evening to colleagues all around the world. It's a real pleasure to be here. My job in the next 10 minutes is to give some background to marine genetic resources in the BBNJ agreement. As Carl Gustav outlined, promoting marine science and building capacity have emerged as two key focal areas in ensuring a holistic approach and bridging marine genetic resources in with the other elements in the BBNJ agreement. So in the next few minutes, I'm going to be outlining a bit of background on what marine genetic resources are, what the benefits are and how they're shared, and highlighting a couple of areas for further discussion, drawing on the draft text of the BBNJ agreement that was released by the President in November 2019. Just changing slides. I'm not able to control this. Thank you. Okay, there we go. Great. So marine life in the global ocean is, of course, biologically and genetically diverse. Marine organisms in the deep and open ocean environments beyond the limits of national jurisdiction are adapted to a range of environments, from the surface of the ocean through the water column, like the twilight zone, to the seabed, be that coral gardens, seabed trenches, or even brine pools. Now, understanding the genetic characteristics and biochemical makeup of marine life can be used in a number of different ways. The first set of applications which usually spring to mind are perhaps associated with biodiscovery when one hears genetic resources. So knowledge, tools and technologies associated with marine genetic resources may be used, for example, in developing new materials inspired by marine life. For example, jellyfish collagen is used in wound healing applications, or perhaps scientific research, industrial processes, or food processes that use enzymes that are adapted to low or high temperatures, or even pharmaceuticals and cosmetics which utilize marine natural products. But there's a whole heap of other types of applications that use genetic knowledge, tools and technologies that are directly relevant to the conservation and sustainable use of biodiversity. And we've really seen this emerging in the BBNJ discussions in recent months and years. For example, genetic knowledge, tools and technologies is used to detect invasive species, to detect illegal, unreported and unregulated trade, or in wildlife forensics. In fisheries management, genetic knowledge is important, and we'll hear more from that from Sheena Talma shortly. In monitoring environmental impacts and in assessing the diversity and distribution and connectivity of marine species, as Dr. Woodall and Dr. Sink will talk about in a little while. So given that genetic knowledge, that genetic science can be used in so many different ways, but not everyone has the capacity to access and utilize that knowledge, those benefits or that science, of course, the BBNJ agreement has been tussling with defining some of these terms. I'll refer you here to the draft agreement for the full breadth of definitions and options, but I want to just here highlight 3 terms and give the essence of what they're likely to mean. The first, of course, is marine genetic material, which is usually taken to mean genes, usually referred to as functional units of heredity. The second term is the one we're concerned with today, marine genetic resources. They're usually defined as genetic material of actual or potential value, but this is open to some interpretation. What is the value? It's not purely economic. It could be ecological, scientific, even cultural, as I've outlined just now. And that's led some to suggest that actually any marine life could arguably be considered marine genetic resource. So it does highlight some considerations for the practical implementation of these measures. Certainly, the utilization of marine genetic resources understandably centres on the conduct of research and development to access the marine environment and to investigate marine life in order to understand it. As I've mentioned, not all have the capacity to do this, and that's why we've really been seeing a great focus on capacity building. I've highlighted here as well the reference to genetic and/or biochemical composition of MGR. That's likely referring to the fact that some natural products, for example, may not fall within the definition of genetic material, and there's certainly been discussion about things like derivatives in other fora. A more straightforward way of really thinking about the solutions to the marine genetic resources issue in the BBNJ negotiations is to look at the potential benefits from genetic resources. Given that science is so central to accessing and researching marine genetic resources, it's understandable that the draft BBNJ agreement relevant to this part, does focus very strongly on access to the outcomes of that research, as well as facilitating the conduct of research itself. I've drawn some examples here from the draft Article 11. I will emphasize these are in square brackets, but it's indicative of some of the discussions. For example, that access to samples or sample collections, like museums or repositories, for example, often referred to as ex situ repositories, would be one potential benefit, perhaps. Sharing of information, either pre or post cruise or research, could be another type of benefit. We've had scientists speak about how useful that might be for coordinating research and for identifying opportunities for participation and capacity building. There's also been some discussion about how this is consistent with best practice, but there's some questions that remain on that issue. A third round of benefits relates to the transfer of technology. And as we know, in the law of the sea context, this is very strongly focused on participation in and access to the outcomes of marine science, like information, knowledge, and data, like skills and training opportunities, cooperation and collaboration, and of course, equipment and facilities, which in this case could range from access to equipment doing marine scientific research to laboratory equipment relating to doing genetic research on shore. Capacity building is another type of benefit that is associated with this type of work. Capacity building could take place from individual human levels to institutional levels, if we think about facilities, right the way through to national, regional, or even global considerations around capacity. Ultimately, science is a global endeavor, and we hear increasingly stories from the scientific community that these types of partnerships are so crucial to global scientific advantage, and is very much a two-way street, not alone a thought of being a one-way donation from one country to another. It's about a partnership. Digital sequencing of information is another type of benefit which is mentioned in the text and has been the subject of some discussion. This was previously referred to sometimes as in silico. We have an expert on our panel today who can help discuss some of the questions that may relate to that type of benefit. And finally, of course, monetary benefits have been mentioned in the discussions, though that's not the focus of my talk today. So how might those benefits be used? Well, in the draft BBNJ text, we see a number of mentions of this, and I've summarized them into 4 key areas. The first is contributing to conservation and sustainable use of biodiversity, perhaps through the acquisition and application of knowledge, as I outlined earlier. The second is promoting scientific research, be that facilitating access to the high seas or promoting the generation of knowledge or technological innovation. Third type of use is in building capacity, both to do science and to apply that science for conservation. The fourth type is in transfer of technology, as I just mentioned. So how exactly to enable this? How to share such benefits? Looking through the draft text, I think of this as being under 4 key areas, 4 key types of approaches, some of which highlight specific mechanisms. The first is participation in research. There's reference to collaboration in science, in collecting samples and data. There's also reference to common funding or pool funding. And there's another reference, of course, to the issue of funding later in the agreement regarding institutional mechanisms. And I would emphasize here as well, this is all in square brackets. I'm giving these examples to indicate the discussions that are underway. Second type of approaches really refers to accessing the outcomes of research, like samples, like data, like knowledge, and the publication of information. I think there's some questions here about how that's going to work in practice and what types of practical mechanisms can really enable that to work. Thirdly, access to information about what kind of research is going on, where it's happening, and who can participate, as well as what's being done with the work. There's mention about rules and guidelines and codes of conduct, which have certainly been proved to be important in other areas and in ocean science overall. We see the role of the clearinghouse mechanism really prominent in these discussions, both concerning marine genetic resources and in related areas for capacity building and technology transfer around access to data, sharing information. There's certainly questions around what that clearinghouse mechanism might look like. How would it involve buy-in from all the different scientists and other stakeholders who would need to be involved in order for it to really work in practice? The provisions relating to capacity building, technology transfer, and other institutional mechanisms, as I've mentioned, clearinghouse, scientific and technical body, funding, are all relevant in thinking about those practical mechanisms. And it's also timely now to really consider how existing aspirations have been translated into practice. We've already seen a lot of aspirations like this in the Law of the Sea Convention, and what types of things we see work really well in practice now, what kinds of scientific partnerships work, and what policy levers are useful to support them. How can we ensure to promote scientific research and enable capacity building? And the example I'm giving on the right here is the idea that just any one of those things may not be that useful. Open access to data may not be that useful without trained people and facilities, as well as the knowledge of networks to access and use it in the first place. So thinking about an enabling environment and the types of mechanisms to create that enabling environment could be really important now. As I close with a couple of slides with further information for things I haven't had time to cover, it's really exciting that we're now going to move on to our other speakers who are going to give their perspectives on those examples and how they're currently working in practice now. I just want to leave you with some references for further information on this. There's some DOSI policy briefs available at this link, which gives some further thoughts on the role of scientific community in benefit sharing around the world. And also there's a series of open access publications here which dive into a lot more detail on some of the issues that I've skirted across in this short introduction. Thank you very much for your attention, and I really look forward to our discussions today.
Thank you very much, Harriet. That was fantastic. Really enjoyed getting that introduction. And I should have pointed out that DOSI is a co-organizer of this event together with IUCN. And this seminar series is sponsored by the government of Sweden. And so thank you very much to our taxpayers. So our next speaker is Dr. Lucy Woodall from Oxford University. She is the research lead for Necton. For those of you who don't know, Necton is a initiative doing a deep water research all across the globe and doing a lot of really exciting research in this field in areas beyond national jurisdiction. So today she will talk about the role of genetics in biodiversity conservation and sustainable use. Over to you, Lucy.
Thank you so much. Good afternoon, good morning, good evening, everyone. So I think for me, I wanted to talk today about just giving a few examples of where we've used genetic resources within conservation and ecology. And after those examples, I'll just give some specific case studies of where I've done that in my own research before explaining where that has taken me in more recent years. So to start off, we all need to remember that there is an estimated of over 2 million marine species, and together the species interact in their specific space and environment to create ecosystems. And as a result of these ecosystems, there's many different benefits to society and the wider planet. These we term ecosystem services. So examples of these services you've heard many times before, but things like half the oxygen produced that's now in the atmosphere is from the ocean. And the fact that marine fish make up about 17% of the protein consumed by humans. So these ecosystems themselves are diverse, and healthy ecosystems are the most diverse. And this is because diverse genetic material within those ecosystems means that there's a greater chance that some individuals will possess the genetic material necessary to recover and resist stresses, therefore adapting to their environmental change. And that is essentially what we have here on the slide. Amazing image of Seychelles, deep ecosystem here, but remembering that diversity allows that opportunity for change. So therefore, it's really important and necessary to document and monitor ecosystems and the species within them. And we know that the genetic resources can help us understand these aspects in more detail. So for example, and I'm going to bring up my next slide now, you can all see. In this slide, what I've done is I've divided all of the different tools into different categories. So samples can be collected, as Harriet's already said, from the natural environment or once they've been removed. This is in situ and ex situ, respectively. I'm going to divide these different samples into 3 different parts, and you can see them here in the table. Now, I'm not going to dwell on the technical aspects of actually what you do and how you analyze these data, but rather I'll detail what useful information could result. And it's key also, I think, at this point to understand that we're not talking about capturing entire organisms or indeed entire populations and communities. And I just want to let you know that I've successfully conducted many studies just using tissue maybe about 1/4 centimetre square and as thin as paper. I think this is important for us to remember when we're thinking about sampling mechanisms. So when we think about individuals in an in situ environment, this could be sampling multiple individuals from the same species within one location. And as here on the table, I've suggested that at that point you can look at population size, identify different mating systems, and this is really important when males and females inhabit different waters across the year. And of course, I've given some other examples as well. So then let's move on to looking at the same species within multiple locations. And from this, we can understand long-term species connectivity, and I've done some of that work myself, and also indicating where sink and source populations may be. And this is incredibly important when we're trying to manage populations. And finally, sampling individuals of multiple different species within the same location will provide more detailed information on species diversity, highlighting areas that are particularly rich or may have a rare combination of species. And again, this is really important for management and conservation. Now moving on to sampling the environment. So traditionally, a lot of work was focused on individuals, and that's how we collected samples. But now many more questions can be answered through sampling environmental DNA, or as we normally term it, eDNA. And this represents things like microbes, as well as shed, secreted and excreted cells, as well as cells from dead organisms. So encompassing the true full ecosystem. Now, it does rely on collecting representative genetic material. Generally, that's done in the water or from sediment samples. And then that information that's collected can be applied to a known library of genetic information. And this kind of disentangles the genetic sequence data that's come from the environment and turn that data into usable information. And you can collect things such as species presence lists. And from environmental DNA, you can also identify the presence of things like invasive species, really important when they're starting to come into an area and hard to see by vision alone. And also things like cryptic species, and those of conservation concern. Now, when we turn to ex situ sampling, I've suggested here, these are samples that are collected within trade, but obviously, as Harriet said, they could also be from museum. And I want to turn your attention now to this specific example of trade and share some examples of my previous work. The first is on seahorses. So starting with this trade, and giving an example of some of my research done through the IUCN SSC Seahorse Pipefish and Sea Dragon Specialist Group. And through genetic analysis, we identified a species, and it's this one right here shown on the screen, Hippocampus hippocampus, the short-snouted seahorse. And we identified this species for the first time in trade. Not only was it important for trade, but we also found specimens had come from outside of their previously reported distribution range. Now, this is really important because all species are listed on the CITES Appendix 2 list, so therefore trade should be documented and managed to ensure that there's no detriment to wild populations. So we had two benefits there from doing one very small study. Next, and very briefly, sediment samples. I collected these in the Southwest Indian Ocean from sediments on seamounts, and we looked at very small worms, nematodes. We discovered that over 90% of these worms were new to science, meaning they hadn't previously been described. And we also found that diversity was really high. Finally, it also appeared that the high diversity areas were really closely linked to the presence of cold water coral reefs that were on these seamounts. So three really important take-home messages from one study. And my final slide before we pass over to my colleague Sheena in Seychelles is explaining kind of how my past research has helped inform what we do now. So I think it's important to recognize that genetic research provides vital information for conservation and management, and that everyone can benefit from this information in different ways. So as Harriet said at the beginning, it's really important for us to work together to make sure that everyone understands what information is possible and we use it to answer our own specific questions. And this is why our Deep Reefs program that is co-produced with Seychelles has an important genetic component that's integral to this work. And this is being used to identify new species of octocoral, to document cryptic species, and also, as Sheena will explain, to determine larval fish identification for conservation and for commercial interest. And this co-production of the research means that it's much better for all. It's not a one-way process, it's very much a two-way dialogue. So for example, in Seychelles, we were able to find the best sites to sample from previous knowledge, and that was both from scientific data, but also from historic oral observations as well. And finally, also it provides a direct opportunity and a pathway to impact. So conducting this novel and research means that it's also useful research. So it's at this point that I'll hand over to Carl Gustav and eventually on to my colleague Sheena, who will elaborate on some of these aspects in Seychelles. Thank you.
Thank you very much indeed, Lucy. That was very illuminating. I think it brings us back to nature in a very exciting way and to science. And I think that's what we're all here to understand a little bit more. How do we deal with managing science? What can we actually get out of it? And what's the common stake in it? And I think we should not forget that understanding what's going on in nature will be half the task to actually get to a point of where we do understand what we need to do and how we can manage it better. Of course, this treaty is all about how do we manage the high seas, and what we're trying to do is find a mechanism that permits us to do a better job on that. And we're very fortunate today to actually have Sheena Talma from also working with Necton, but she's also at Rhodes University, where she is a professor and of the Institute of Aquatic Biodiversity. And she is actually, I think, a good example of someone who's worked closely both with international cruises and the type of high seas research that we're talking about here, but also in a developing country setting. And she's currently based in Seychelles. So she's close to the field when it comes to operating the types of questions that we are trying to address through our treaty. So today, she will give us a little bit of an idea of the genetic research perspective from Seychelles. So over to you, Sheena. And we need to see you as well. There you are. And we can't hear you, so you need to put your mute button. Let's see if we can you hear me? Yes, but they're very weak. So maybe you can speak up a little bit there. So we.
All right. So thank you so much for that introduction and for the promotion. I'm unfortunately not yet a professor. So I was studying at Rhodes University and very fortunate to have studied there for quite a long time. Like we were chatting, I'll be talking a little bit about genetic resources and genetic studies within the Seychelles. I just can't move the slide.
You need to be a little bit closer to the microphone. We still can't hear you very well. Maybe you can.
Can you? Is that better?
Yeah, but you really need to scream into it, I'm afraid. Sorry about that.
That's all right.
And we give you.
Any better now?
Yes, thank you.
Perfect. All right. So I can't control the slideshow yet. There we go. Perfect. Yeah, so I'll be talking about the genetic research perspectives from Seychelles. And just to clarify, although I'd love to be a professor, I'm not there quite yet. And I did study at Rhodes University for quite a long time. So I'll be talking a little bit about the research that I was involved in that all happened within the Southwestern Indian Ocean. So just to go back to Lucy's slide and understanding how my research links into that. So firstly, I used samples for the two studies I'll be presenting. The first one, we collected samples in situ and also ex situ. So we collected samples by fishermen at islands and also from museums. Okay, so there was a mix of samples coming from all over the place. So the first study I'm going to look at is the genetic connectivity of bonefish. So basically, we wanted to find out, are these fish really well connected within the Southwestern Indian Ocean? Okay, so bonefish are like luxury tourists. They like to live in very clear water, next to bays, and generally on coralline islands. Okay, so really you think of the tropics when you think of bonefish. So why are they important? Well, for a very long time, the whole world thought that this species was actually just one global species, and the ones that you would catch in the USA would be the same ones that you would catch in the Indian Ocean. But after using genetics, that's when we actually found out that, hey, there are more than 12 species that we know of, okay? So because they're all very similar, it's very difficult to identify them just using their morphology, so just using what they look like. So we wanted to find out, are there different species that live within the Southwest Indian Ocean, one, and two, how closely related are these species? So to do that, those were our two questions. So we went out and collected samples. So we partnered up with organizations that were fishing in these remote parts of the Seychelles Islands, and as well as Mauritius. Okay, so that's the only places that we could get our samples from. That's where we focused on. And at the end of it, we collected quite a lot of samples. Now, because we wanted to answer two different questions, we used two different types of genetic methodologies. There are lots of different types of genetic studies and genetic methodologies. So the first, to answer one of our questions as to what type of bonefish we find in the region, what we did is we used the genetics that we inherit from our mom. Okay, from all our mothers, we inherit some type of genetic material. That's what we used. Then we used a more advanced platform, which is next generation sequencing, to kind of find out how related they were. Are they cousins? Are they sisters? Sort of like that kind of thinking. So next, what did we find out? Okay, so we found out that Firstly, there are two species of bonefish that occur within the southwestern Indian Ocean. So what we did find is one of the species seems to like the flat islands, the coralline islands, whereas the other species can live in granitic islands, so islands with high mountains. Why that is, and a lot more studies need to go into it to actually see whether this is because of the structure of these islands. Okay. So the other thing we found out to answer our second question was that they're actually really well mixed. So throughout the region where we were looking at, the Seychelles and Mauritius, they were actually all closely related. And this graph shows you that. So if they were different, they would have been clustered out separately, okay, in their own individual groups. However, you can see on this graph that they all actually clustered quite closely. So all one population. This has very important implications for management. If they were separate populations, we would put in different management strategies as opposed to when they're one mix throughout an oceanic region. Now, the second study I'll be talking about very briefly, and this is one that I was involved with Necton and a lot of different organizations in the Seychelles and partners overseas, was looking at what kind of fish, baby fish and eggs we find throughout the Seychelles waters. So again, back to Lucy's table, we did in situ sampling, which means that we took samples over a period of time when we were out on a cruise. Okay, so we covered the area around the Seychelles. So we went all the way down to all the island groups within the Seychelles Islands. Okay, so what we do is put a net through the water, get a whole bunch of plankton, which are small organisms that live throughout the water column. And then we removed just the fish and the fish eggs because you get a whole bunch of different type of organisms. Okay, so why, you might ask, don't we just look at the fish and the fish eggs and say what they are? Well, because that requires a lot of expertise. Okay, so what we wanted to do is try and evaluate or have a first kind of snapshot of what kind of fish and fish eggs we find in this region, which is why we're using genetics to sequence the fish and the fish eggs and compare it to a library in the web that will tell us what they are. So what can we learn from both of these projects? Okay, so project 1, which looked at bonefish connectivity, and project 2, which looked at eggs and larvae. The first project was cross-boundary. It was Seychelles and Mauritius. The second one was within a country's boundaries. But for me, the most important lessons is that partnerships are really, really important. Collaborations are really important. That's how you make a project successful. But more than anything else, they have to be long-term partnerships and collaborations, as I myself have been fortunate to have had. So for my genetic studies, for example, I had the opportunity to learn new skills, to write papers, to be part of every process of the science involved. So all of these really valuable lessons need to be applied towards any initiative towards the BBNJ. Thank you very much.
Thank you very much indeed, Sheena. That was really riveting, and having worked and spent a lot of time in Seychelles, I'm always delighted when I get to see it, particularly this little sort of group of islands out there. I think that's off Paradise Beach in Pralan. But in any event, I think you did a splendid job. I could hear you behind the dog there. We were having a little bit of competition at one point, but I think it all worked out fine. You did a splendid job, so thank you very much indeed. So we now turn to our last speaker, but before we do that, I also wanted to give you all the encouragement to put in questions. We're getting some good questions in here, and I will try to assign them to different parts of our panel. I'll introduce the panel after our last speaker. So our last speaker is Professor Kerry Sink. She's with the South African National Biodiversity Institute, where she's a research associate. And she's also at the Institute for Coastal and Marine Research at the Nelson Mandela University. So Kerry will be telling us a little bit about deep ocean research capacity considerations, a perspective from South Africa. Kerry, you have the floor.
Hi, everyone. Thank you very much. Let me see if I can get control of these slides. Oh, there we go. I really appreciate this opportunity to share with you some of my experience. And today I'm really going to draw from 3 areas of work that I've been doing in South Africa. The first one is our National Biodiversity Assessment. The second one is the recent development of a new network of marine protected areas. And thirdly, dedicated capacity development efforts in deep sea research. South Africa assesses its state of biodiversity every 7 years, and we do this across all realms. I've been leading the last 2 marine assessments, and we do this at 3 levels, for ecosystems, for species, for genetics. We also pay attention to the benefits of biodiversity, and we put this all together. In the case of the recent assessment, this is a 700-page report. It has contributions from 70 authors, and it allows us to give a bird's eye view of the state of biodiversity. And this is used to inform decision-making across multiple sectors. At the ecosystem level, we have excellent frameworks for reporting on the state of our marine ecosystems. We've recently moved to the IUCN methodology for assessing ecosystem threat status, and using our national map of marine ecosystem types overlaying our protected areas, we can report on ecosystem protection level. At the species level, we can use the IUCN framework for reporting on the state of our species. In the marine realm, we've got the highest levels of data deficiency across all realms, and taxonomy is one of the major impediments we have in assessing the state of our species. And if we could strengthen our integrative taxonomy and apply molecular tools, we'd be in a much better position to describe, assess our marine biodiversity at the species level. However, it's our genetic chapter that is actually the weakest chapter and the more neglected chapter. This component of biodiversity, although we're able to report on the risks and so show some impacts from fisheries, for example, on a couple of commercially important species, we don't really have the temporal data sets, the trends in time and proper indicators to track the genetic level of biodiversity. And these knowledge gaps and capacity limitations really compromise our ability to report on our state of biodiversity. What we need is some kind of framework that could strategically track trends in genetic diversity. And to develop this kind of framework, really it shouldn't be something that South Africa tries to do on its own. We'd really benefit from international collaborations to do this. And this is one of the key elements where capacity building could really make a difference. In our benefits chapter, we found that South Africa's ability to derive benefits from its biodiversity is actually limited by these knowledge gaps and technical challenges, particularly in deep water. From our science-based assessment, we develop a suite of priority actions, and there's 2 that I'd really like to reflect on here that are relevant. The first one really shows the need to overcome barriers to strengthen our bioinformatics and molecular approaches. So we're limited in our assessment of marine biodiversity. And as you've heard from Sheena and Lucy, if we could apply molecular approaches and strengthen our bioinformatics, we could really do this more quickly, more cheaply, and using less invasive techniques. The second one really speaks to the integration and modernization of our data systems. And the key elements here are to improve our bioinformatics and better integrate genomics in this. So if negotiations can support the kind of actions that they can do this would really make a difference to South Africa. The second area of experience that I'm drawing from is our work to design marine protected areas. South Africa proclaimed 20 new marine protected areas after 13 years of work last year, taking us from 0.5% of the ocean space around South Africa to 5.4%, and we're still striving to get to 10%. One of the limitations of this work has been an absence of especially deep water data to support connectivity. We know for marine protected areas to work well that they need to be effectively connected. So you need to incorporate migration data, but also population genetics. And to do this properly, we don't just need to look inside our own waters, but for example, in the seamounts, we'd also need to understand connectivity to the seamounts in the high seas area. And that's the relevance of some of this work, even at a national scale. I've been working with many young scientists and students and colleagues to actively develop capacity for offshore research in South Africa. And so I'd like to just share a couple of examples of what's been working well in this aspect. In 2011, South Africa got its first remotely operated vehicle to enable offshore research. And this vehicle's got a depth limit of 300 meters, although we don't have the cable to get that deep. And it has an underwater claw, so you can take samples. But the problem is we don't have any system to store those samples. So each time you pick something up, you see something that might be a new species, or you want to do a genetic study, you pick it up, you have to travel through the Agulhas current, one of the strongest currents in the world, to try and get that specimen to the surface. This is not how deep sea research should be done. So we need that technological innovation to sort this out for our system. In 2011 and 2012, South African scientists started taking us into deeper water, and through the systems like this tow camera system that belongs to the South African Environmental Observation Network, we've been able to extend our reach to see ecosystems in up to 700 meters of water. But the challenge is we can't take any samples, and without being able to take any samples, we can't describe the incredible biodiversity we see. And this is frustrating. South Africa is a country with 3 oceans, the Indian, the Atlantic, and the Southern Ocean, and we've got incredible biodiversity. It's estimated to be the 3rd highest endemism in the world. And so we need to get the capacity into the hands of the people who have this diversity in their waters. And we can do this by inviting developing country scientists to meaningfully collaborate and participate in research. It's still really demotivating when I talk to people from developing countries about challenges in deep sea research, and they show me that still in 2020 there are scientists who publish papers from the deep sea in developing country waters without any collaboration from anyone from not only the country, but even the continent where that work is taking place. So these are some of the barriers that we experience. The financial and technological barriers are pretty well understood and well established. Being able to get a vessel offshore and into deep water. The piece of equipment I have there is a device that actually our ROV pilot built. It's a GoPro in a housing that got the first footage in South Africa's slope at 1,000 meters. And we call it the chiropractor. It's quite a problematic piece of equipment, but we really need to be able to extend our reach into deeper water to study our waters. It's not just the financial and technical barriers, but there are also academic barriers. You know, most of my professors were esteemed intertidal ecologists, and there's limited capacity in universities to help students who are interested in the deep sea. And so this is something that I really think that we need to change. And then in terms of any opportunity for experience and awareness, like being able to showcase deep sea biodiversity is important. A further challenge that we face is that often grants, we have to compete with scientists who have access to very sophisticated equipment and have a long established research efforts. They know what their species are because they've been able to do collections. It's really difficult to publish your work in a high quality journal if you aren't able to identify the organisms that you're looking at. This applies to the publication process too. Some of the things that have really been helpful in enabling our work in strengthening capacity in the deep sea have been some of the academic champions from other countries who've collaborated with us. In the yellow overall in that picture, you see Lisa Levin from DOSI, who I met at a deep sea conference. And Lisa really inspired a whole generation of marine scientists in South Africa and Namibia in a single sabbatical, by coming and spending time here, giving guest lectures and working with us. Importantly, when we went to sea and Lisa came with us, this really gave us an opportunity for someone experienced to be lending that experience to us in deep water. And so she reached many of us in the field work, and in that way, she was able to help us overcome our constraints. So really more people learned many more things than had one South African travel to an international, to participate in an international expedition where they might have the opportunity to, or access, or the chance to learn about some deep sea equipment that they might not actually be able to work with. Increasingly, there are projects and programs that are developing some of these collaborations. Lucy and Sheena shared an excellent example, and we are starting to develop some of these collaborations in the One Ocean Hub project that I'm involved with. And what really helps there is when scientists are willing to slow down and understand things from our needs, to understand the technological limitations and work with the strengths of local people. I'd also like to say that any opportunities to give people experience and awareness of these things. I've got Jodie Oliver, who's going to be a PhD student working with us for some new molecular work on the call here with us today. And it's really important to give young scientists the opportunity to start working here. On the right here, you can see Kirsty McQuaid, who's a South African scientist who overcame our academic challenges through collaborations with Kerry Howell at Plymouth University. And as she's just graduated with her PhD in deep sea ecology, I think she's probably South Africa's first deep sea scientist. And you can see her here at the International Seabed Authority meetings where she's been making a difference. The other kind of critically enabling factors, and I think areas where the agreement could really make a difference, are in mechanisms that can help us to strengthen offshore and genetic research, to help developing countries work with developed countries to co-develop research. And by funding and incentivizing collaborations, many countries could actually benefit. Some of the very practical things that helped us were funding for postgraduate studies, the opportunity to travel to international conferences or participate in conferences, and even things like journal subscriptions. I think that the methods need to really be practical and simple. I think developing countries, many of them know what their needs are, and these need to be effectively communicated. I think approaches that will encourage collaboration and participation, that's the kind of collaboration and participation that we need for understanding and managing and benefiting from and protecting the high seas. those kind of collaborations and partnerships will also be incredibly beneficial to developing countries. Thank you very much.
Thank you very much indeed, Carrie. That was very interesting, and I think really illustrates many of the key points here in terms of how do we make this work for all parties. And you know, the whole underlying issue, I think, is to find mutual benefit. How can we create a system that permits everyone to have as much interest and say, and where the collaboration is done in such a way that we get good science, we also get trained people all around, and we further understanding. And in these days where we see a lot of conflict and strife around the world, I think it's extra important to find mechanism for collaboration so that we can all move forward in a more harmonious existence. So we've now gotten quite a few interesting questions, but I would really encourage you all to put in new questions and ideas that you would like to have our very illustrious set of speakers and panelists describe. And let me now ask the panelists to put on their videos, and I will introduce them as they come along. First on here is Marcel Gaspars. He is professor in chemistry at the University of Aberdeen in Scotland. He's been working on this area of marine biodiscovery for more than 25 years. He's also been providing scientific advice for the BBNJ process since 2014, and he has included in the the IUCN delegation to the BBNJ negotiations in New York. And thank you very much, Marcel, for agreeing to be with us. Then we have Abby Brown, who is professor of intellectual property law at the University of Aberdeen as well. And she has been involved in BBNJ processes since 2018 and has also been inspired to become a paddy driver. So that's very exciting and something that I personally also enjoy doing a lot. And then we had Judith Gobin, who's the first female professor of science at the University of the West Indies, at Augustine campus in its 60-year history, which I think is something we should really celebrate in Trinidad and Tobago. And her expertise is in marine biology and marine biodiversity in coastal and shallow environments, as well as the deep sea. And then finally, we have, I think, Lucy, and we have Jody Karin Olivier. And I must admit, I missed your bio information here, so you'll have to tell us yourself a little bit of your affiliation. Apologies for that. So Jody, over to you. Don't we hear you, Jody? Jody.
Hi, sorry. Hi, I'm currently a research assistant at Stanby under the supervision of Professor Kerry Sink. So I have some experience in marine genetics, and I will hopefully be doing a PhD next year on some deep sea genetic work.
Wonderful, welcome. We're happy to have you here. So I think the first question that we got in was on the topic of prior or post notification of cruises and research. Now, this is a topic that's been discussed a bit within the framework of the treaty. And then we would like perhaps to start out with asking Marcel, but maybe some of the other would also want to address this on. What do you see as the issues here? I mean, you've been out on a number of cruises. How useful is it to have these prior notification or post notification, and what kind of text would be enabling to help us get to a good solution? Marcel.
Thank you very much. Hopefully you can hear me. So the idea behind prior notification of a cruise is for a number of reasons. The first one is that other scientists might be able to join that cruise and take part in the research as it takes place. Or if that's not possible, they might be able to direct some of the research that takes place on the cruise. For instance, they might say, I would like some samples from these locations for my particular research. And most importantly, I think it prevents overlap of cruises doing the same thing in the same place from different countries or from different cruise operators. So that's important to remember that part. And it means that, you know, if this information is made available a month ahead of the cruise departing, you might be able to get better coordination between cruises and their scientific goals. And if you have post-cruise notification, what it means is you update that information, what samples were actually collected, you know, did you manage to meet the cruise objectives? And normally the cruise report will file that information anyway, so that information exists. It needs to be shared in a coherent format in a sensible way. And it means at the end of it all, you have data in a format that is useful to scientists around the globe. And you might also be able to tell people where the materials are at that time, who is taking possession of them in terms of research, and where the data is located. So that would be my feeling for being able to share data and information ahead and after the cruise.
Thank you very much, Marcel. Anyone else want to chip in on this one? If not, I think we'll move over to the next question. So a number of people have highlighted the importance of philanthropy and the private sector in terms of science, and how can we move forward. So one of the questions for, I think, all of you is, you know, what is the good way for scientists and universities to build capacity on genetic research and analysis of marine biology, and how can we use private individuals, for example, shipping companies, to address this type of topic? Anyone who would like to say anything? Jody, for example?
If it's okay, I can jump in here.
Go ahead.
Yeah. Okay. So from small island territory as Trinidad and Tobago, as we all are aware, islands are very special, with a really, really large ocean space compared to our land space. And we have, I can say that Trinidad and Tobago has benefited from philanthropy in the form of the EV Nautilus coming to the Caribbean. It was actually the first expedition, although exploration rather than full research. It was the first exploration vessel where we had local scientists, myself, and on the second leg, Dr. Diva Ammon, who is a well-known deep sea biologist as well. And just to add a pin there, that she is the first deep sea biologist, first and only purely deep sea biologist for Trinidad and Tobago. And with myself, the sort of biologist hanging on and getting into deep sea biology after the introduction on the EV Nautilus. So we are very pleased that this type of philanthropy exists. And at the moment, I'm also fortunate to be on the Rev Ocean Scientific Committee. And it's amazing. Those are amazing opportunities for small island territories and less developed countries. And we really, that's really what we need to be able to. It improves and increases the network for us and where we can actually meet people and work with real scientists on board. As was mentioned earlier by Kerry, I think it's a really good idea. That's what we have to do. It's to continue that partnership and collaborations. And at the moment, philanthropy does help us a little bit more in our territory than the oil and gas explorers, exploration companies rather, because at the moment they're interested in getting out there, doing the job, and there's always that health and safety issue. So it does work for people like us, and I'm really pleased that there's more philanthropists coming on board. It can only be helpful for us. Thank you.
Thank you very much. Does anyone else have some questions on this that you would like to share, or ideas about this private philanthropy that you want to share?
I may come in if I can. Abby, can you hear me?
Yes.
So maybe approaching from a slightly different angle, if one involves the private sector particularly more, and I totally support the contributions made by colleagues who are much closer to this than I am about the ongoing value of this. One of the issues I'm very interested in is intellectual property. Now, if we are having the private sector involved, then the private sector may want to get a patent, or it may want to, when it chooses to publish the information, it may indeed not wish to publish that information. It may wish to say that it is secret, or it may wish to argue that that information, particularly in particular structures in a database, is a subject of an intellectual property right. Now, intellectual property rights are extremely good things, but they do give a private power. So I think it's very important that one is aware of that. And one of my suggestions in the contribution I'm trying to make in the negotiations is we're trying to work out that the treaty should consider engaging with IP rights, that we should think about, should there be some limits, which is quite appropriate from an IP law perspective, should they be considered, so we don't get to perhaps an unfortunate situation when valuable information is with the private sector. They think that's appropriate because they made that investment, which is a fairly reasonable approach. And as such, it isn't being as shared and isn't as having as maximal an impact on, say, capacity building. So I think that's sort of my main contribution I want to make, always to ensure that we are having those conversations. There is no one clear answer, but I think that the more we can have those conversations, I think that's of great value, so we can reach a solution which will work for everyone. Thank you.
Thank you. That one relates also, I think, very much to what is the difference between using this type of information for an economic purpose versus doing it for scientific research. And I think that is a very interesting sort of angle on this. So anyone else want to express any views on that?
I can say a little bit more on that if you
Sure. Yes, please.
Weird, like just that often I think it can be quite difficult to draw a line between the two. Work might start off at the moment. I know a tremendous amount of work was going on in relation to COVID-19. Work might start off from a purely scientific point of view, if one likes, and then suddenly one might be trying to get something to market, then that might become more commercial. So to try to put a clear line behind that and in between the two can be difficult. Marcel, who I know is on the call, is just an example of many scientists who have moved into the commercialization, the company spin-out phase. So the two extremes, of course, are very different, but in the middle, I think it can be difficult. So I know there's a strong view that one should try and avoid reference such as commercial use or non-commercial use, or at least be very clear, perhaps the underpinning issues which one is trying to address.
Well, of course, you might start out as doing basic science, and then gradually one discovers an economic potential, and so it can be very hard to see at which point you need to make that transition. So I think, you know, how do you share this type of information? How do we put this in the treaty? I think it'll be a continuous challenge for us. Right, so then we also had another question here about how common access to samples should work in practice. You know, what's the best way of sharing these type of samples, and you know, what kind of mechanisms can we put in place that's practical and, you know, doable, and also not too cumbersome. So again, sharing the benefits of the science, but creating a system where we actually mutually move forward rather than, you know, to exclusively do so. Who would like to say something about that? Lucy, maybe?
Hi, thanks. Yeah, I can start off. So I think from my experience, actually making sure that, you know, we've got a diversity of voices around the table and co-producing that science is really important. So I think that's the first thing. It shouldn't start with once the samples are on board. It needs to start pre-expedition to really understand what that value is and what that value is for everyone on the ship, but also for people who can't get on the ship, because of course we realise that often there are birth limitations. So I think that's the first aspect. I mean, I would say that you know, obviously right now, I guess, you know, the good analogies have been some of the work that we've been doing in some of the EEZs, because obviously that's where there is already some regulation, and actually we've been very successful being able to have those conversations, talking to all of the stakeholders, so that there is a real understanding of the samples that might come in and the value of them to the different parties. I would say alongside that, it's really important to make sure that information is shared in a way that is accessible to all. It's all very well to be able to produce the information, say, well, it's in a database somewhere, but actually having access for everybody in some random database, you know, isn't necessarily always very easy. So yeah, I mean, this goes beyond just that point of samples, I think, into the larger philosophy of how we're doing the science and why we're doing the science, and the conversations we're having around our specific questions of interest, and where we want that information to go. So I see it as a much larger point of the philosophy of what we're doing.
Thank you very much, Lucy. Very helpful. Anyone else who want to chip in there a little bit? Yeah, Marcel? You're muted.
Yeah, I would agree with Lucy. It's a really good point to say, you know, it's about the planning of the research and knowing that we're doing research with common goals for the right reasons, and that we know where the data goes in the end. And I think that can be helped a lot, and it's addressing one of the questions is by the right kind of data standards. Make sure that we have at least a minimal data set of things that we're collecting, and that goes into the relevant databases so it can be found again, it can be accessed, and it can be reused by other people as well. The question then also came about traceability and how you might use a system that is, I would feel, a light touch system rather than a heavy-handed full track and trace system that traces something through discovery to commercialization. So do all the good science that we need to do. So make sure we have the standards in place, we put the data where it should be, and it's interconnected. But also make sure that at the end of it all, we can find out where that sample came from if we do have a commercial use based on that. Because it may be 20 years from now and it may be a broken chain, it may not be a single, you know, linear process, it may be convoluted, but there is still a way for the final commercial user to trace it back to its origin, perhaps using a unique identifier.
Thank you very much. So then we also had a question a little bit about how long we should have embargo periods for primary research. You know, of course, we all know that as scientists, you want to publish several different papers when you go on a research cruise, and you might not be able to do so as quickly as you would like to. So how do we see this thing and balance that towards, you know, the sharing of scientific data to ensure we move on? And anyone want to address that a little bit? Carrie, maybe? Or anyone else?
No, I mean, I could speak again, but maybe you're sick of hearing from me.
No, not at all. Go ahead. We didn't have many speaker slots, so I think we should try to assert ourselves in the true male way, right?
No, just kidding. No, so the idea behind the embargo is indeed that a person who may have directed that research deserves the right to first use that material as long as they're working on it. What is important to recognize as well is that there may be possible other non-competing uses for the same material as long as there's sufficient materials. And a good example of that is the Norwegian biobank called Marbank, which allows this non-competing use, so that they have a material ex situ in a database, in the marine biobank. Then you can ask to use that, but if somebody else is already using it for the same purposes, you won't be given access. But if there's sufficient material and you're using it for a non-competing use, then the research is still possible. So I think that's a good way to think about it. But if the sample is extremely limited and you have it for a specific reason, preserved in a particular way, then I guess there should be an embargo on that so that you use it. And it might be an updatable embargo. It might be after 3 months, you ask, are you still working on this? But that could be very complex to manage. But something like that. And although at the moment, the movement is away from embargoes for data, for sure. So data should be open access right from the very beginning. Whereas for materials, you may want to consider an embargo. You might want to consider materials and data to be treated separately.
Thank you. So we have another thing here, another question. What are the main points of divergence between states in the negotiations of marine genetic resources that could compromise the BBNJ adoption? And I think maybe this is something that, Abbe, you would want to address? Or maybe Harriet?
Sure. Oh, can you hear me? Harriet, I don't know if you want to come in first, because you can perhaps give a wider contribution, and then I could come up on the IP side. I think there's a note from Carla as well, which ties in with that.
Sure. Do you hear me okay?
Yep.
Great. Hi, thank you very much. I'm going to sort of highlight just a couple of examples, I think, of where those key points are. I think firstly, with regards to it's linked in some ways to the provisions relating to capacity building and technology transfer, which is this question of sort of voluntary and mandatory types of commitments. I think that we've sort of had a lot of discussion on those points in recent months, and I think one of the things that we can really draw on for from the discussions we've heard from panelists today is that there's already quite a lot going on both sort of voluntary and mandatory bases, and there's probably a case for both of those working together, perhaps at different levels. A second issue, I think, has been regarding the specific mechanisms for sharing benefits and the extent to which there's a role for the agreement in actually developing such mechanisms. I would say that the discussions, again, that we're having today and elsewhere regarding existing practices do highlight there's certainly a role for new mechanisms, as well as strengthening existing mechanisms to enable those outcomes that we're discussing in terms of both science promotion and capacity building. I think there's also been some issues relating to whether or not promoting science is at odds with achieving those capacity building objectives. My personal view on this is that it absolutely is not. The provisions regarding conditional freedom of research in the high seas are very much consistent with the types of information sharing and capacity building and inclusivity discussions that are going on. So I think that there are sort of a couple of areas, but we can certainly see plenty of material for bridge building on those. Thank you.
Thank you, Harriet. Abby.
Great, thank you. So I think intellectual property is a point of which there is a variety of views. One angle, I think, is the fact that if one interferes with intellectual property, that is a strong disincentive to private sector investment, a strong disincentive to scientists being rewarded for their activity. I think there's a lot of different views which tie into that. I think another view which very much ties in with the point that Carla has raised is, well, intellectual property shouldn't live in this agreement because it lives properly somewhere else. And my view is that's not quite accurate. IP certainly does live somewhere else. There's the Intellectual Property Agreement, which some people may know as TRIPS, in the World Trade Organization agreement, and it does set out standards by which countries have to protect intellectual property. But it also provides various optional exceptions, and I think it's certainly open to states, and I would certainly suggest that states exploring those exceptions in relation to BBNJ. The other place, so fragmentation is not new to the IP debate. Where IP lives is in the World Intellectual Property Organization, and it has been doing a lot of work for quite a long time in relation to traditional knowledge and genetic resources. So I think there's 2 points that come from that. One is it's been work going on for a very long time. It hasn't really been particularly effective. It's also really focusing only on one of the IP issues, which I think can be relevant, the controversial, the important issue of disclosure of origin. But it hasn't reached an agreement. And I think if this is considered important to the BBNJ process, it would be valuable to think of addressing it within this process. Also, there is the fact that the most recent WIPO drafts are suggesting it's not going to look at the area beyond national jurisdiction. So one can think that it should be explored elsewhere, but it's not being explored elsewhere. And I think if we do think this is of value, it should be done in the BBNJ process. It's been suggested to me, perhaps with some fairness, that the BBNJ process shouldn't be solving the problems of IP, and I would totally agree with that. But my suggestion is that if we are seeing IP as possibly blocking some of the outputs of effective technology transfer, capacity building, for example, then we should be working appropriately within IP to ensure that the BBNJ goals can be pursued. And I think there's a lot of middle ground. The dialogue, I think, is really moving on, that there can be a measured solution which can enable the wider agreement to be delivered and also to be fully workable.
Thank you very much, Abby. That was very interesting. So we had another question here about common standards for data collection and sampling. So basically, how can we set out various types of protocols that would permit us to know in what format we would get things in? Now, I think this is always a tricky one, because as scientists, we're always constantly pushing the boundaries of coming up with new methods of measuring things and discovering things. And that might mean that we don't conform with the same sampling protocols that we used earlier. So anyone in our panel wanting to address this issue? Just put your video and sound on and I will know that you're there. Lucy,
Hi.
there you are.
Hi. Yeah, so I think, I mean, obviously people like Marcel have done a lot of work on this, but I think something for us to think about is it's not just the standards, it's how we compare those things. Because as you said, you know, sometimes we don't conform to standards for a particular reason because we're asking a very specific question that requires some very specific data. However, you know, there are a lot of initiatives trying to think about what are those key aspects of data collection that can be that can be standardized. But then thinking about when we do our own research, what are other data that we can collect at the same time that might help put the data that's being collected at that moment within the context of data that's been collected historically and will be collected in the future. So I think, you know, it's more than just standards. Again, it's thinking about this in a whole, thinking about historic and future needs as well.
Thank you. I have another question for Harriet here. What is your view with regards to fish and its derivatives as marine genetic resources? So of course the main question here is, we do a lot of fishing all over the place. Should that then be considered actually doing genetic sampling? And, you know, how would you put provisions in for that? Or should there be big exclusions for all kinds of different types of organisms that aren't considered because they're used for other commercial purposes? So Harriet, do you have some reflections on this?
Thanks, Carl-Gustaf. I seem to have video again. Very, very briefly, I think suffice to say that this issue has been discussed at length as reflected in the draft BBNJ agreement. What I would say about it is that, as we've heard today, genetic knowledge is useful for scientific research to advance the process of science, but is also useful for management. Whether or not the provisions of the BBNJ agreement would interact with fisheries management, I'm not going to speak about that. What I will say is that advancing that knowledge, building capacity to acquire and utilize genetic science is useful in many different ways. I think that's the really crucial thing to focus on here, is the opportunity that presents itself to actually build scientific and technical capacity through this agreement. Thank you. I think Marcel will speak as well.
Marcel, yes, please, go ahead.
I just wanted to say that if you are considering it, I mean, there are things you can do with marine genetic resources in terms of fish, and that would be just to look at the genomes and analyze those for, I'd say, something like antifreeze proteins, which you find in your ice cream to make it taste more smooth, that kind of thing. So these things exist already, and people are already doing that work. So, but that doesn't require you to go back and then harvest a whole load of fish. You're just looking at the genome to do that. So I think that's very separate from fish as a resource compared to fish as a genetic resource. So it becomes a two-sided question. I mean, in reality, I could go to the supermarket right now, get a piece of fish, analyze its genome, and work with that. I don't need to go fishing a long way away, and it only requires a very small amount of actual material to do that work. So it's a good question to ask, and I think the two should be kept separate. Fish as a commodity versus fish as a genetic resource, but fish should be included because of the potential value of the genetic resource there.
So when I have you there, Marcel, why don't I also take the next question for you? So there's a question regarding the resistance to requiring pre-cruise notification. So from your side, how common, normal is it to do pre-cruise notifications and post-cruise reporting? And what can be the exceptions for that?
So I think the most normal thing is that you apply for a cruise through a national funding body, such as in the UK, the National Environment Research Council, and that would be done years in advance of time, and your cruise path would be planned as part of that cruise application. We'd indicate what kind of sampling gear you would need, and if it's funded, you would then find out whether or not the materials that you need are on board, the right kind of sampling equipment, and you would go through the cruise path to make sure you're not accessing any restricted areas, such as marine protected areas. And post-cruise notification is required at that point, and it's put in the data portal of the National Oceanographic Centre in Southampton. So that data is always reported straight afterwards. That is not the same for every country, but I know that in the UK and Japan, those systems exist and are very, very robust.
Great. Another question we got is relation to intellectual property. Do you think these issues should stay within the new draft instrument, or should they be better handled by an organization such as, for example, WIPO? I think the question here is, how specific is the high seas, and is it useful to have that internally in the treaty, or should you refer to another body that might already be managing this in a reasonable manner? So Abby, over to you.
Sure. Yeah, I think this builds a bit on some of the points I made before. It could be addressed elsewhere, but it isn't being addressed elsewhere, and the international politics of IP move slowly as they do in the BBNJ space. So I think if there are particular solutions that we really do want for BBNJ, then we should look at exploring here. I can say with quite a lot of confidence that what could be done in the BBNJ space would not be inconsistent with what is permitted under the TRIPS agreement, because it is rather draconian powers, and broadly what is being discussed in relation to WIPO, both in the genetic resources treaties and also some developments once seen, for example, in books and visually impaired treaties. There's a trend towards some exceptions being increasingly embraced to IP rights. So it's a view which is very commonly held, that IP should not be discussed here, that IP should reach another fora. But if it is felt that a particular solution would work for BBNJ, then I think it would be useful to think about trying to create one here rather than hoping that someone else will.
So we also have a question in, Abby, for you regarding COVID-19, and if there's anything that we've learned in terms of best practices that would be relevant for the negotiations of the BBNJ.
Yes, it's an interesting one, that. Well, I do remember when I first met many of you a couple of years ago in Aberdeen, and I would talk about things like, oh, AIDS drugs, and people didn't share AIDS drugs, and there'd been problems with synthetic biology. And I think people looked at me and they didn't quite believe me that big companies might not want to share their technology to meet an important public good. And COVID-19 has given rise to lots of debate on this point. We have seen a couple of companies which spring to mind, and I hope I have them right, hope no one's going to sue me, but I think, for example, Gilead and Sanofi have been some which have been mentioned as perhaps looking at taking a more traditional approach to developing a solution to getting a patent, to making lots of money from it. And there's been a lot of challenge to that. Some countries, again, I think Canada and Chile are particular examples, and lots and lots of activist pressure for saying no, countries should be passing laws to say that if you do develop a cure for COVID, you will share that much more widely with everyone. And that's that type of solution which has a lot of resonance with quite a lot of the sharing, the commons-based approaches which we are seeing explored here. We have also seen exploring yet another international organisation which can be relevant. The World Health Organization has been doing a lot of advocacy and a lot of initiatives on, say, technology pools in relation to COVID-related medicines. So we're seeing a bit of a wave to a more the place where perhaps a lot of the BBNJ debate tends to naturally live, a view based on sharing, based on collaboration rather than the private sector. But it's interesting from the IP side to see the IP world having to really engage with that and maybe starting to lose. I tend to see things a bit in fights rather often, it's my Glaswegian background, but IP often wins this fight, and IP is maybe beginning to lose this fight within the COVID space. The communal approach is maybe starting to win, and that might be really interesting if one wishes to continue to make those arguments in the BBNJ space.
Thank you so much, Abby. So I think we've had a very rich discussion, and we could go on for a long time with this. I can see that already. We're getting lots of good questions in, and apologies for all the people who put in good questions, which I didn't have a chance to addressed to our panel and our speakers. Now, I just wanted to give the speakers a final opportunity to make any short final points, because in fact, our time is already up. And I also see a few of our attendees have already dropped off. So just, 30 seconds of final thoughts that you would like to give to people. And again, I would say we will send out a little questionnaire at the end of this. So you have an opportunity to give us some advice if you want us to do further work on this thing. And then again, I would like to thank everyone who tuned in today and for your very active participation. And of course, particularly thank our panelists and our speakers. So please put on your cameras also so I can see who wants to say. The only one I see is Marcel so far, and Carrie, who wants to say a couple of last words? Sheena, do you want to go? You're still muted, I'm afraid.
Can you hear me now? Is that better?
Yep. Go for it.
Okay. Thank you so much for having us on the call. And I think I'd really like to take some points from Carrie's talk. Thank you so much for presenting that. And yes, whatever we do, it really needs to be inclusive, needs to ensure that there are these great partnerships that last over a longer period of time, not just, a 2-day workshop or whatever. And there are opportunities there, and it's great to see that this conversation exists where these things can be put in place early on. So thank you very much.
Thank you. Carrie.
Just to echo Sheena's comments, I think that the opportunities for collaboration and for working together can really benefit developing countries. And thank you for giving us the space to share our perspectives. Thank you.
Welcome. Lucy.
Yeah, I think I just like to say that, we have demonstrated that genetic resources are really important for management and conservation of our oceans, and they are going to be vital in allowing us to understand the best opportunities moving forward. And this can only be done with meaningful engagement that is allowing that mutually beneficial outcome.
Marcel.
I just wanted to pick up the point that Abby had made, which is about the COVID crisis. From a scientific point of view, I've never seen such rapid and open collaborations taking place, based both around free access to data and free access to materials to further research. So, I've been involved in research projects that have involved the UK, Portugal, Israel, South Africa, Philippines, and things like that, very, very quickly putting together projects addressing the ways that we can find treatments for these diseases using marine resources. So again, it really is important that we embrace that more open future in science.
Thank you. Judith.
Just really pleased about the inclusion of SIDS in the discussion. And my hope is that it's going to continue after the negotiations and after we have an instrument. It is very important for us to be included and to have that sustained collaboration, partnerships encouraged across the board, because it doesn't end here with the negotiations. It really only begins for us in being able to sustain the collaboration and the partnerships and the work to improve the science in SIDS territories. Thank you.
Well, thank you. And we all know that SIDS are actually great ocean nations and not small island nations. So, and with great minds is the other thing. So again, congratulations on your appointment. Abby.
Thank you very much.
I just like to thank everyone. I think it's been a fantastic dialogue, very multifaceted, very interdisciplinary, very broad geographically, and fantastic to see such a wide range of people who have been contributing in their different ways. So thank you all.
Thank you. And Harriet, please go ahead and sum up a little bit. Sorry about that. Please go ahead and sum up a little bit. Sorry about that.
Thank you so much, Colga and stuff. It's been a fantastic discussion. I was reflecting how much the conversation has changed around marine genetic resources since I started doing my PhD on this issue about 5 years ago or so. We spoke very much about the commercial potential And I think today's discussion about the conservation applications relating to the study of genetic diversity and the opportunities to really think critically about how we can build capacity in a meaningful and long-lasting way is really exciting and really promising that in the coming months, as we move towards the fourth and final round of negotiations, we can really achieve something spectacular. So thank you so much for convening this webinar and hearing from such a great round of speakers. Thank you so much.
Thank you very much, Harriet. And I wanted to thank Dozy very much also for the collaboration on this. Without all your hard work, particularly Harriet and Christina Gjerde have been tremendous support, as well as Aurélie Spadon from our own team here. I also wanted again to thank the Swedish government for their generous support to enable us to do this work. So without further ado, I wanted to end this here and say we will have further discussions on environmental impact assessment and strategic environmental impact assessment, and we'll be doing that one in a couple of weeks' time. Then we'll do another one which is on area-based management tools. And finally, we're planning to do one beginning of July, which would be more on the financial mechanisms for the treaty, as well as governance aspects. All, I think, are riveting and super interesting questions, and I think on top of everyone's mind these days. So we look forward to these further discussions. Any ideas for improvements we would greatly appreciate. So all of the attendees, you are more than welcome to actually send us some comments as we send out a little questionnaire at the end of this. Thank you very much, and looking forward to next opportunity for us to interact. Goodbye.